Now next we come to the ultrastructure of cilia and flagella. Before that we need to see that how this flagella is embedded in the cell, because we talked about that it is present on the free surface. So this is a simple representation of the cell. This is the outer surface of the cell, bacterial cell we have taken into consideration over here, and this flagellum is getting embedded into this part. This part has been magnified, which you see over here, that the flagella is basically how it is embedded, that there is a hook, there is a filament and there is a basal body which embeds the flagellum into the cell envelope of the bacterial cell. As you can see over here, this is the outer membrane that we call as glycocalyx in the prokaryotic cell, and this happens to be the cell wall, followed by the plasma membrane. As you can see over here, there are two rods. Through and through a rod passes through the sleeve. The sleeve is basically the basal part which gets embedded into the cell envelope, and there is a rod through and through which is part of the filament itself, which subtends till the plasma membrane. This is the point of understanding that you have to understand, basically, that how a prokaryotic or a eukaryotic flagellum for the case, how it gets embedded in the cell envelope of a given cell and cell membrane or cell wall in case of other cells. So this is basically, this entire portion is the filament, which would be the longer part that we see in the flagellum. This is the filament. It is having a hook for enabling the whip-like movement of the flagellum. This hook is followed by the basal part, which has a sleeve and a rod which enables the fixture of the flagellum into the cell membrane and the cell wall. Next, what we deal with is that how this flagellum and cilium, supposedly this is the flagellum or the cilia which I am talking about. I cut it from here and then I observe it from this angle under a microscope, in an electron microscope. So we get to see this ultrastructure that you can see over here. Before going to the ultrastructure, you need to remember that there is a basal part in flagellum which differs from the exoneme, okay, in its ultrastructure. So what is exoneme? Come back, we have this part. This part is basically exoneme, which would be surrounded by plasma membrane, and there would be a basal part which would be different in ultrastructure from the exoneme. Coming back to the ultrastructure, we have this exoneme which is having 9+2 arrangement of microtubules which have actin-like filaments. You know what microtubules are, what is their function. So microtubules are basically polymers which get intertwined. We have actin filaments over here which get intertwined and form the microtubules, and there, because they are hollow from inside, you can see over here this hollow part is being represented. So we have 9+2 arrangement in exoneme, while in the basal part it is 9+0. Second thing you have to remember is that over here this is a micro 9 which is represented by microtubule doublet, okay, and in case of basal part it is microtubule triplet, and there is no central part, okay, central part is not having the pair of microtubules, it is triplet. So we need to learn the structure of exoneme basically in detail. The basal part can be skipped off because this is what is the main structure which is responsible for bringing about the movement of the flagellum. Now what you have to remember is that there are 9 pairs of microtubules which are joined to each other through nexin, which enables the dynein to be controlled from taking over the digestion of all these microtubules. Okay, the part you have to remember about nexin is that it joins the microtubule pairs. You see a central microtubule pair which is having a central sheath around it, and to this central sheath from each doublet the A tubule subtends a radial spoke which is in contact with this central part. Alright, I am going to repeat it again. That we are having microtubule doublets which are in close association with each other. With every movement, we have these microtubules which are moving. Now we'll see how that happens, but for once you are having 9 pairs of microtubules, each tubule named as A tubule and B tubule, A tubule and B tubule, you can remember it that way. From A tubule subtends the dynein arms. Dynein is a protein which is responsible for bringing about the movement which ultimately leads to the cell movement by the agency of flagellum and cilia. There are dynein arms and there is an axon which joins each microtubule to each other so that there is a coordinated movement. Apart from that, the A tubule, microtubule, it also subtends a radial spoke which is in close contact with the central microtubule's central sheath. There is a pair of microtubules in the center as well, and there is a central sheath to which each of these radial spokes joins. Now you have to remember that how the movement is brought about. Please cram this diagram by heart because it is very important for understanding the function of the flagella, which is movement. Now you know how the movement is to be brought about. You see dynein arms. Dynein arms are having nothing else but dynein protein. Dynein is used for breaking down the ATP. Alright, this dynein would bring about ATP hydrolysis. So ATP would come into the scenario, it would break down in the presence of dynein, and what would happen is that both the microtubules A and B, they would slide over each other. When that dynein breaks down, the ATP breaks down, it brings about the movement of each microtubule over the other. When there would be movement, that is sliding movement of microtubules on each other, there would be entirely coordinated movement in this entire exoneme, and that would be responsible for bringing about the movement of the flagellum or the cilia, because the ultrastructure for both of them is similar. It is the dynein arms and the microtubules in close proximity to each other which bring about the movement in the axoneme in total, so that the cell can move, it can show movement which was whip-like movement as we observed in the case of flagellum and cilia.